Agartala, Andhra Pradesh. He has his research group WRG, that is Waste from Aquatic and Terrestrial Environment towards Resource Recovery. This research group is mainly aims to develop processes for treating industrial waste from lab scale to industrial scale and further focusing on producing valuable added products from industrial and domestic waste. This group is passionate about developing a sustainable solution for water pollution, moving towards a circular economy. The research expertise of the group are in the design of experiments for various water treatment processes, including bioprocess engineering, bioremediation, advanced oxidation and reduction process, and also ecological studies. Sir works with national and international parties from various institutes and research centers on water pollution and related issues and research management. He also received his ongoing research grants, 4 grants from Department of Biotechnology, from Government of India, Department of Science and Technology, and from Ministry of Earth Science. And also he completed 2 research grants from Board of Nuclear Science and BART, Institution of Engineers, Kerala State Council for Science and Technology and Environment. He received several awards such as Young Engineer Award 2021 to 22 from Environmental Engineering, IEI Kolkata, and received Young Scientist Research Award from Baba Atomic Research Center, Mumbai, and also received his Young Scientist Research Fellowship from Indian Academy of Science and also from TNSCST. And we have various publications such as 60 with cumulative impact factor 200 plus 12 book chapters and also 23 national and international conferences. His proactive venture in research paved path for us to collaborate overseas like University of Paris, France, and Cranfield University, UK, and Shanghai Marine Resource and Environment Research Institute, China, Institute of Physical Chemistry, Polish Academy of Science, Poland, and also and at national level in IIT Varanasi, IIT Guwahati, NIT Calicut, and Kerala Veterinary and Animal Science University, Kerala. And he is also a member in a professional body like community member in the American Chemical Society and a life member in International Water Association and a member in an Indian Institute of Engineers and also a life member of International Association of Engineers. I take this opportunity to welcome you, sir, and thank you. Session over to you, sir.
Yeah, thank you, Vanaja and Dr. Amal. I believe my voice is audible and screen is clear.
Yes, sir.
Yeah.
You are audible, sir.
Fine, thank you. So, good evening to everyone, all the renowned faculty members of various institutions and research scholars, and my dear students also. And this is a part of ATAL program where the topic is about revolutionizing the healthcare and biotechnology, the role of AI and computational techniques, where the positive sides of pharmaceutical compounds and how to enrich the opportunities in healthcare technology or healthcare systems is we will be addressing. And the entire ATAL program is focused on that. And I'll be the one of the person who will be standing odd man out in this particular area, where we will be, our team is working on degrading the antibiotics. Okay, so we will take you to a different arena of pharmaceutical compounds and what are all the impacts of pharmaceutical compounds if it is not managed very well. So that is the focus of this particular session. And I would like to thank Dr. Amal and their team, his team, for giving this opportunity. And I would like to thank ACT ATAL for this opportunity too. So I'll be focusing for another 1 hour of a time. I will be focusing on the various sustainable technologies in PPCPs, where I will be focusing mainly on the antibiotics. And we will try to see what are all 3 to 4 different case studies also, which focuses on these degradation. Yeah. Yeah. So the first point is about the water for peace. So everyone knows that the water is a very important entity which is available all throughout the world, but we couldn't able to get a safe water. for drinking purpose, and it extends from all different living systems, not only for humans. It extends to animal systems also, for the plants also. The quality of our water is not up to the level, and about 844 million people lack basic water services, while 2.1 billion people lack clean, safe water on their living premises. And if you take about the agricultural sector, accounts for about 69% of the freshwater use, and 19% of the freshwater use are relates with the household systems. And the demand of water, you can able to see here with respect to the comparison between the years of a decade. Here you can able to see everywhere the water demand is keeps on increasing irrespective of all the continents. And when it on the other side, if you come into the picture, when you think about the sanitation and hygiene, in 2015, about 2.3 billion people lacked basic sanitation services, while 4.5 billion lacked a managed sanitation service involving the safe treatment and disposal of the sewage. And still 900 million people are still practicing the open defecation. Why we are giving more importance to open defecation system is this is one of the important route by which the unmetabolized antibiotics or pharmaceutical compounds released into the open environmental system. So this is where we need to target how we can able to reduce these concentration of these antibiotics or pharmaceutical compounds through the approach towards effluent treatment plants or sewage treatment plants like that. Okay. Yep. Yeah. So getting into the system of pollutants, there are three major categories of pollutants we can able to group it by World Health Organization. So one is the pollutants of poverty, and which is the sanitation related bacteria and trash and litter in waterways, where 80% of the world's wastewater is released into the environment without treatment. So it is about the pollutant of poverty, where the underdeveloped countries or the countries which are below the poverty line and which are declared as if from the World Health Organization belongs into the class of pollutants of poverty, where the sanitation related issues will be a dominant one, where we can, in those places we cannot able to expect the emerging contaminants in a larger picture, where the lot of pollutants which are concerned and directly related with poverty will be higher and rate. The second one will comes about the developing countries, where the pollutants of growing prosperity, so the industrial sectors will be very high. So the chemicals and waste from the industries and agriculture will be done in the terms of a business. So the fertilizers and pesticides will be in a large scale picture. So these comes under the pollutants of a growing prosperity, and the emerging pollutants comes with the developed countries, where the emerging pollutants in such a way that the different types of plastics in from micro to nano scale or pico scale, and different pharmaceutical drugs, personal care products, where and these perfluoro compounds like PFAS, and many different compounds are coming up now. And recently it has been declared as the e-waste is going to create a very big havoc in another period of 10 years of a time, where everyone are switching over towards the electrical system, electronics and electrical systems. So the dependence of heavy metals and the rare earth metals will be very high. So these are all the classes which comes under the emerging pollutants, and which is explained as it is an emerging pollutants comes under the micropollutant class, which ranges from the level of a nanogram per liter to microgram per liter. So due to its low concentration, it is very complicated to detect and analyze. So if it is a higher concentration, we have a robust methodology to measure what is the concentration of these pollutants. But since it is a lower concentration, our methodology is what we are practicing now in wastewater treatment plants or in the detection systems will not be sufficient enough to measure these pollutant concentration. So this will naturally get accumulated inside our body, and slowly it gets magnifies, and it creates a lot of toxicological damage into the living system, not only to the humans. It starts from the zooplankton and phytoplanktons, and it extends to the level of human beings. So that is the magnified version of the toxicological profiling with respect to the micropollutants. And out of these, many different types are there. Industrial chemicals is one class, microplastics, steroid hormones, pesticides, personal care products, and pharmaceuticals. So throughout this particular session, we are going to target on this pharmaceuticals, the personal care products, as well as the steroid hormones. So these three are the major class of emerging pollutants which are prevalent in most of the countries. Irrespective of the poverty level, most of the countries are dominant in experiencing these pharmaceutical and steroid hormones in their effluents. So what about the environmental concerns about the wastewater pollutants? So unsafe water kills more people than the disaster and conflicts. This is a very interesting information and alarming information also. The natural disasters is creating around 8,200. Due to conflicts, it is around 87,400. But unsafe water and its related diseases will create a very big damage to the human system. And you can see here the share of people without access to basic drinking water service in 2020. Here you can able to see this is the region which you can connect with the earlier slide, the regions of poverty, the regions of developing countries and the developed countries. So you can able to correlate very easily with this particular map. And what about the impacts of water pollution? Yes, everyone knows about it creates a very big demand for the oxygen with respect to biology as well as with respect to the chemical degradation. So this is an unexplored area in most of the research works or in most of the wastewater treatment plants. We will be neglecting on these particular nitrogen compound. Everyone will be targeting on the carbon, how to reduce the carbon in terms of total organic carbon or inorganic carbon. But there is another important emerging contaminant which is called nitrates, phosphor and sulfur, which many people are neglecting in their wastewater treatment plants. But this is having a very important potential to create a stunted growth in the children. And another important area will be the salinity because of the use of extensive salts in many different plants. Like you can take the textile industries where a lot of salts are getting used, and you can able to see the aquaculture systems where the salinity is going to maintain. So they are adding artificial salts are getting added into the normal fresh water. So these releases create a very good impact and a very considerable impact in the water. So that ultimates creates with respect to the pollution. And here in this particular context, we are going to deal only about the drug pollution. And now we are not going to deal about the pesticides. We are not going to deal about heavy metals, or we are not going to deal about the textile dyes or leather industry. We are going to target on the drug, pharmaceutical compounds, antibiotics. So what about the environmental concerns of PPCPs, especially antibiotics? The major concerns is the misuse of the antibiotics. One simple example I can able to give you for the benefit of the undergraduate or postgraduate scholars who is present in this meeting. And if a doctor prescribes an antibiotic for a period of 5 days of a time to reduce the fever or any kind of an illness, and once after taking 2 days of an antibiotic, naturally there will be a control of the fever. So naturally what we will think is, oh, our fever got reduced, so I will stop taking the antibiotics. This is one of the important criteria by which you are developing your microorganism which is present inside the system is developing the resistance against these antibiotics. This is one of the very important serious issue which has given by the World Health Organization during 2019 until date. Okay. Now, apart from antibacterial resistance, now antifungal resistance is also coming up. So the misuse of antibiotics is a very important issue. Improper disposal of the antibiotics, which we need to take care very, very much. Now the government of India and almost all the state governments are taking much important care how to handle the biomedical waste. Earlier time it was not there. We were just dumping in the landfills. So through leaching process, this will enter into the groundwater, and through our consumption, it will enter into our body, and our systems get resistant towards these kind of pharmaceutical compounds. But now, entire country as well different states are taking a very good initiatives to manage the biomedical waste in a much proper way. So what is the importance of it? To identify what is a point and non-point source. Point source means from which source the water is getting contaminated. So which will be it can be easily correlated with the industrial components like pharmaceutical industries, those who are producing the pharmaceutical compounds. With the improper management of their industrial effluent will release these partially metabolized or unmetabolized or complete antibiotics or pharmaceutical compounds into the water. So that in turn affects the aquatic ecology as well as the human terrestrial ecology also. Non-point sources, we cannot able to identify what is the source of this particular thing, but the entire river is completely contaminated with pharmaceutical compounds. So we have to have a tracking procedure to identify the point and non-point sources, where a very big lack of research is there. Third important component is about the lack of robust analytical detection. As I said earlier, these pharmaceutical compounds will be in a very lesser quantity, and these are quickly transforms from one particular structure to another structure. So the reactivity is very high. So what is the problem is we know what is the compound, but once it changes its structure from a different form, we couldn't able to identify what compound it is existing. So that is a very important gap we are facing in the detection of pharmaceutical compounds. And the fourth important domain where a good amount of research is still expected is how to analyze the toxicity profiling inside the system. So we know very well if you consume a 10 milligram of a particular tablet, we know what is the effect inside our body. But unknowingly, we are drinking a water which contains a lot of antibiotics. If it keeps on accumulating inside your body, what happens to your body system? So that ecological side or toxicological side, ranging from lower organisms to higher organisms, including humans, is still a very big lacuna is there. So these are the four major areas which we have to focus so that the antibiotic pollution can be reduced much better way. So the treatment technologies for the pollutant removal, I can put it in a comparative way. So here you can see the existing method where the detection and the treatment system in most of the countries you can tell. And on the other side, we have to be focused on the future. So this is the existing practice what we have in most of the countries, and preferably in developed countries, this practice is little bit advanced. So we will be analyzing the conventional pH, COD, BOD, TDS, total dissolved solids, total suspended solids or total solids, alkalinity, acidity in the wastewater treatment plants. And regarding this, all the treatment procedures will be focused. Whether we will be going for sedimentation or we will be going for aerobic treatment or anaerobic treatment or sludge separation, RO treatment. And this is how the existing procedures are there. But we have to be focused on the future through a detection in the treatment system where these emerging pollutants has to be sensed. We have to understand how much amount of pesticide is there in the water, how much amount of antibiotics is there, how much is the dyes are there or detergents are there. Depending on these important micropollutants, your treatment procedures has to be developed. So you can able to see here the micropollutant detection and the special treatment procedures has to be incorporated with the existing practice so that the treatment will be much more effective to control these micropollutants in a much better way. So this is one of the important focus of our research group. And the sustainable technologies in PPCPs like antibiotic degradation. Yes, you can. Everyone in this particular session is well experienced about the biodegradation. We can use a microorganism to control a particular pollutant. Like if you take textile dyes, you can use azoreductase enzyme to degrade the azo dyes. You can use, if it is a pesticides, the pesticide degrading enzymes can be used. So if it is a lipids rich environment, oil waste, you can use lipase enzyme producing bacteria to control it. So you know when you are very well used to this. But one of the important challenge and one of the important setback of using this microorganism in this antibiotic degradation is if you start to use the microorganisms to control or degrade these antibiotics, slowly you are releasing the antibiotic resistant bacteria into the system. It will not be done in a single day. You will be executing this work over a period of 1 year or a 2 year of time. Gradually, these antibiotics will also get released into the environment where the natural microbiota develops resistance against this antibiotic. But many researchers are using these microorganisms to control these antibiotics, but it is not advisable as per the World Health Organization guidelines. So we are switching over to alternate methods where the physical methods or chemical methods can be employed. So now we are into the advanced oxidation process where we are trying to couple one or two different methods based on its own advantage. So whatever may be the advanced oxidation process you are going to do, there are many different types like photocatalysis, ozonation, electrochemical phenton, or microwave and sonication. There are many different advanced oxidation processes are there where whatever may be the process, our ultimate aim is to produce these free radicals. So the magic of free radicals in eliminating pollutants is enormous. One important magical molecule is called hydroxy radical. Many different molecules are there. Superoxide radicals are there. Ozone radical is there, and sulfate radicals are there. So these are having a very good oxidation potential, which can remove these pharmaceutical pollutants in a very shorter time. So it will not the half-life of these radicals are very high, very less. So what happens? This is very quickly reactive, and it is a broad spectrum. It is not like a enzyme and substrate reaction, like very specific. These are all broad spectrum. Whatever may be the compound you are giving to this radical, it will degrade into smaller monomers. So that is the advantage of these radicals. So our ultimate aim is to overproduce this radical so that we can able to degrade these antibiotics or any kind of a pharmaceutical pollutants in a very shorter period of time. So that is what one of the important areas where we are working on towards remedial purpose towards PPCPs. So coming to our water research group, we are working on waste from aquatic and terrestrial environment towards resource recovery, and our aim is to work on towards the One Health principles, where we are targeting on the human health, animal health, and the environmental health. So there are to carry out with this particular session, out of these antibiotics, we are going to target a compound which is called fluoroquinolones. There are 4 different generations of fluoroquinolones existing in this world as of now. The first generation is nalidixic acid and siloxacin. Second generation is afloxacin and ciprofloxacin. Third generation is parfloxacin and gripafloxacin. Fourth is moxifloxacin and gemifloxacin. And these are all the 4 different antibiotics we are having as of now. And one of the important alarming signal is in most of the states of India has started to prescribe the 4th generation of antibiotics, which means our body as well as the many different microorganisms started to develop resistance against first generation, second generation, third generation also. Now many doctors have started to give 4th generation of antibiotics to control the multi-drug resistant diseases. Okay, so what happens if the bacteria started to develop the resistance against the 4th generation? We don't have any other antibiotic to prescribe under the fluoroquinolone class. So, and in the first session, you might have got a clear experience that to develop a drug, it takes more than a 5 to 6 years of a time. Earlier, it was more than 10 years of a time. Now, due to the advancements in the bioinformatics and many different drug discovery tools, now it can be possible within a 6 years of a time. But still, it is very short than the resistant developed by the microbes. So what are all the routes by which it is spreading across the different system? It is spreading through human activities. It is also spreading through veterinary, because in order to control the microbial infection in the veterinary systems, we are also using antibiotics. So through its excreta, through its urine, through the human excreta, these unmetabolized antibiotics are getting released into the system, and this creates lots and lots of adverse effects into the human life, as well as any life of the any different ecosystem you can able to take. There it creates a very big damage. So let us start with for the first case study, which we started to working on this particular system on 2017, and this is the use of ionizing radiation, like a high energy gamma radiation, to degrade the antibiotics. This particular work is funded by Baba Atomic Research Center, where a team of researchers we are working on it, and here we used the gamma radiation, which comes under the ionizing radiation. So this gamma ray, when it passes into the water, it releases lots and lots of radicals. So to name an active radical species, the free electrons will be released, hydroxy radical will be released, hydrogen ion will be released, that is hydronium ion, and H2O2 will also be released. So this is the method by which the attack over the pharmaceutical compounds has happened. So this is called indirect reaction. The second one is directly it will target the bonds of the antibiotics. So this is when the gamma rays pass into water, the water get ionized and the radicals will be generated, and these radicals will take care of the degradation. This is indirect reaction. The direct reaction is directly it will target the bonds which are present in the any kind of a pollutants. Let us take here as a pharmaceutical compounds, and the degradation will be taking place. And this is the structure of the afloxacin, which belongs to the second generation of fluoroquinolone class. So what we have here, you can see the clear picture how the as the gamma ray dose increases, it has started to the compound has started to degrade, and it reaches a very close to the plane level of systems. So here we have tried to optimize, we have tried to identify what are all the factors which affects this system. And interestingly, the pH plays a very crucial role, and dose rate also plays a very crucial role, and the concentration of antibiotic plays a crucial role. And additionally, we try to use the electrons. As you know, the 4 different radicals are used in this, prepared in this particular water, through water. Only OH radicals was used for the degradation system earlier. But in this study, we started to use these electron also. So we could able to increase the rate of a reaction by almost double the time. By how we have done, we have additionally added one catalyst which is called hydrogen peroxide, which taken care of the continuous removal of this particular continuous production of the hydroxy radicals, so which taken care the degradation in a much faster rate. So these are all the four major factors which we have developed in the system, and this particular work has been absorbed in the Baba Atomic Research Center, and it is in the pilot scale application level in a water treatment plant in Gujarat. Yeah. So the case study number 2, we started, okay, let us think about the gamma radiation. Yes, but what are all the disadvantages of the gamma radiation? What we felt is we couldn't able to put it in a decentralized system. So there are two different types of treatment system. One is called a centralized treatment system and decentralized treatment system. So decentralized treatment systems is good, which means we can treat our own wastewater from a house or the apartment point of view. Centralized means there will be a collecting treatment plant where we will be sending all the effluent into the system. They will be treating and they will be sending back the fresh water to us or to any industries. So one of the important challenge with respect to the gamma radiation is we cannot able to incorporate in a decentralized system. So we wanted to try any one method or to develop a method which can be used in a every household systems. So we wanted to check the electrocatalytic removal, where we try to incorporate the use of electrodes, very specifically active as well as passive. Initially, we started with the passive electrodes, then we have moved to active electrodes, and now we have started into the BDD electrodes, where the sludge formation will not be there. So this work has been published in Environmental Research Journal, and here you can able to see this is a kind of a two electrodes which we have developed. This electrode has been developed from our side, and which is having a very good potential to generate the radicals, OH radicals. And here the anode is there, and here the cathode is there, right? And we have added the pharmaceutical compound containing this water into the system, that is nothing but the fluoxetine, and we started to give the electric current into the system, by which the degradation, how far the degradation is happening, we wanted to try. And since we earlier, the first time when we have used the passive electrodes, a lot of sludge was also generated, and we wanted to characterize what kind of a sludge it is creating, because we wanted to move towards a sustainable point of view. Because if you are putting in a household system, we cannot able to give another procedure for treating the sludge. So we wanted to give a compact procedure where what is a component of the sludge, so that whether these sludges can be reusable or not. So that is what the whole idea about this electrochemical oxidation system. And interestingly, we found this is how the roughness of the cathode has changed after the use of the system. And we interestingly found out that through the XRT analysis of the sludge, we got an interesting information that two important minerals are present in these two different colored sludge. One colored sludge is red and another one is the green colored one. And where the red colored one is mapping with the mineral name of a gupite, and the green one is mapping with the mineral name of a lepidocrosite. So which is nothing but the both are the components of the ferrous. So the ferrous containing sludge is very important for activating the Fenton's process also. So that is why the another important factors has come into the play. If you see here, the initially we targeted for the electrode based treatment, but when these sludges has created an additional advantage to carry out the electrophenton process also inside the system. Though it is not our target of study, the presence of these sludges, ferrous based sludges, has created another advantage of electrophenton based systems. So this is one another area where you can able to pitch it. So now we have started to work on utilizing the what modification in the electrodes and the different types of catalyst we are working on now with the nano hybrid catalyst, so which enhances the rate of reaction much faster, by which the sludge cannot be generated. So that is another important area where you can able to pitch in. And the case study number 3, it is like adsorption. So it is a very important area where the what we can tell. It is very importantly applicable in the Western Ghats or in the Northeast. So since I positioned in Northeastern region now, we wanted to see how, because decentralized treatment is not at all possible in the Northeastern region. So we wanted to work how to, sorry, centralized one. So we wanted to move towards a decentralized one. So we wanted to make it how the adsorption can be used so that in each house they can able to use it. So here the bamboo is very predominantly present. So we wanted to try how these bamboo biochar is able to quench the fluoroquinolones, not in a single one, multi-component system. We wanted to try many different antibiotics and through a biochar which we have prepared. So what we have done is we have collected the bamboo gum and we have converted into biochar, and the conventional characterization we have taken, and we wanted to study in a single component system as well in the multi-component system. So in a single component system, the different parameters, as you know, what are the importance of a contact, how much we have to interact between the adsorbent and adsorbate, what is the importance of PZZ, or how to make it very easy for the pupil without having a much intense facilities, how we can able to make it in a very small scale system. So that is what our idea behind this development. And further we have to think in a professional way also, we have incorporated the adsorption kinetics and conventional isotherms to see how the efficiency works. And but here the ultimate aim is to prepare a material which can be used for the regular household purpose. That is what the sustainable use of these bamboo biochar is the objective of this work. But to put it in a more technical way, we have studied the different kinetics also in the system. So this is one of another important area where you can able to pitch in because these antibiotics can be recovered and desorbed back. So which can be used, which can be put it in a very smaller environment, and you can go for the treatment. One advantage of this adsorption is this adsorption will reduce the volume of the effluent. So it will try to concentrate the pollutants. Then you can treat the highly concentrated pollutant through any kind of an advanced oxidation process. So that is the ultimate aim why we have incorporated adsorption. Yes. So here you can able to see the different types of the different color shows the different class of antibiotics, one on afloxacin, levofloxacin, and ciprofloxacin. And interestingly, the process parameters are also works very well. And with respect to the temperature, it is working very well at a higher temperatures also. So the conventional, the kinetic parameters, we started to work on it, and the ciprofloxacin is having a very good absorption power with respect to 44 milligram per gram of a bamboo biochar. And the conventional isotherms we have developed on it. And in a multi-component system also, it worked very well. And you can able to see the effect of a pH where it works very well with respect to the base pH of a 5, then it to the neutral and towards the alkaline. Since the compounds are with respect to the PA value and pKa values of these antibiotics, the acidic range is found to be very interesting towards the PZZ value of the bamboo biochar also. And here, with respect to the total increased concentration, you can able to see from this graph up to 400, 450, as well the 500 milligram per liter is efficiently removing these kind of the mixed components of the pollutants. And the fourth case study, with this we will conclude, and it will be a little detailed one, and it is a very important area where the sulfate radical based advanced oxidation process is comes into the picture over here. Caffeine is one of the important pharmaceutical and personal care products which are not only present in the coffee which we are consuming, which is also present in most of the drug medications. And here I have listed down some important characteristic features of the caffeine, where it is highly soluble in water, and the half-life in water is about 10 years of time. So you can able to imagine the half-life is more than 10 years. It is having a very good point to get accumulated inside your system. Okay, so we need a very robust radical which can degrade this particular caffeine in a very shorter period of time. So that is how we wanted to work on the other radical, which is called sulfate radical. So here you can able to see the amount of caffeine per cup. A normal cup which we consume, it's around 3 milligram, and the brewed coffee or instant coffee is getting up to 100 milligram of caffeine. You please remember this number in your mind. Later we will switch on to the toxicological profiling. You will understand how it is creating a damage to the zebrafish. And the sulfate radicals, so here we wanted to work on this sulfate radical, which can be activated by the persulfate. It is having a very good redox potential, and it can be activated by many different routes: heat, temperature, sonication, UV, ozone. Many different methods are there to activate the sulfate radical, and it is have a higher half-life than the OH radical. So one of the best advantage of the sulfate radical is it can be activated in a multiple route, and you can able to control the process also. In hydroxy radicals, it is not possible to control, but in sulfate radical you can able to control. So the caffeine, you can able to see certain images which where the caffeine deposition is there very high in the marine systems also, and in the sea sponges and in the marine macroalgae. You can able to see how far these caffeine are getting settled. And the toxicity damage we wanted to study, so we have used the zebrafish for assessing the toxicity damage, because 70% of the human genome and is matching up with the zebrafish, and 82% of potential human disease related genes are also present in the zebrafish. So that is an interesting phenomena with respect to zebrafish. So we wanted to try with zebrafish. So you can remember this magical number, 95 milligram. Okay, so here you can able to see the toxicity profiling. So this is what the overall graphical abstract of the work. Yeah, just a minute. Yeah. So the caffeine toxicity we wanted to study in the zebrafish. We wanted to optimize the process. We want to analyze the end product. So this is what the overall picture, which we are going to see in another 10 to 20 minutes of time. Okay. So here you can able to see the developed embryo in a E3 medium, which is the medium where we have used to grow the zebrafish. And here you can able to see the 5A, which is a control, which doesn't have any kind of caffeine. 6A to D, 6A, 6B, 6C, and 6D, which contains a 200 milligram per liter of a caffeine. So you can able to see the effect of toxicity the caffeine is creating in the healthy embryo developmental stage of a zebrafish, right? So this is the healthier one, which will be in a straight line kind of a stuff. When the caffeine is added into the system, you can able to see a kind of a bend. Okay, and here you can able to see the kind of a coagulation over here at the higher concentration and higher volume of the system. Okay, but here in the bottom 7A, 7B, 7C, 7D, you can see the much higher concentration, 1000 milligram per liter of a caffeine. Okay, and here you can see the complete coagulation of the complete embryo. So the thing is here, and on the other side, I have also said that the 75% of the genes are having human orthologs for the zebrafish. So the same kind of an effect, the caffeine can also incorporate into the nervous system as well as in the different developmental stages of the human beings too, right? Yeah. Yeah. So now, yeah. So now here we wanted to try how the caffeine is stable. So you may think that whether the caffeine can be degraded in a different pH or in a different temperature or in a different time interval. Here you can see from the ABC and ABC graphs, you can able to see the caffeine is highly stable in different pH. There is no change in the spectrum, and there is no change in the structure of the caffeine molecule even at 80 degrees Celsius. So that is why when we are boiling the coffee, the caffeine is highly stable. That is why we are getting used to it. So it is highly stable at a different temperature up to 100 degrees Celsius. That's what we know from our day-to-day consumption of coffee, right? And here only at the persulfate we tried, there is no degradation. But when the sulfate radical is getting activated at a temperature of 80 degrees Celsius, you can able to see the decrease in the concentration of caffeine. So this is what one of the important hypotheses we want to put forth. By use of the persulfate, you can able to degrade the caffeine molecule within 60 minutes of time at a concentration of more than 100 milligrams per liter. So that made us to evaluate how these can be studied to get a clear picture about different types of concentration or a different time interval and different kind of parameters we want to work on. So here you can able to see we wanted to study the effect of temperature on this. So here we have tried with the three different temperatures. For your better understanding, you can see the rate constant alone here. Right, so at a 40 degrees Celsius, there no reaction was happened, so the rate of reaction is very almost zero. But at 60 degree, the rate started to increase. But at 80 degree, it reached the maximum up to close to 6 ppm per minute. A 6 ppm of a caffeine can be degraded within a minute at this particular rate. You can able to establish the process. So you can able to imagine how much quantum of caffeine you can able to degrade by using this sulfate radical based process. Yeah, the second one is about the effect of a pH. So this is a very crucial one. So because we wanted to understand, we wanted to take up this to an industrial level where we are in touch with one particular industry, which is a pharmaceutical industry, which adds the caffeine compound in their medications in a much higher quantum. So they have said that they are operating the entire process at extreme pH. They will be operating at pH 11 or 12, which are highly alkaline and a higher acidic range. So we wanted to try how these caffeine can be degraded or how these radicals behave at a different pH. So that is what this is the stoichiometric equation behind this radicals at a different alkaline pH as well as the acid pH. So here you can able to see at alkaline pH, the domination of hydroxy radicals is very high. So that is why the pH 11 stays at a much higher rate, and which is evident from the rate of a reaction also. Further, the next acid range is much higher because the disulfide radicals are also very strong in degrading these kind of a pollutants at a much faster rate. So which we wanted to experience how these processes behaving at a different pH with respect to different operational industries and with respect to the concentration of the caffeine. So that is where we tried with a different concentration of the caffeine, how far you can able to reach. So here you can able to see the rate of reaction is almost 7.6 ppm at a persulfate concentration of 10 millimolar, and the rate is also at 4. So above 4, the rate of reaction is almost more or less same as 7, 7, 7 and 7.6. So there is not much differences there. But still, if we wanted to take the higher concentration of degradation, so we preferred the highest concentration as 10 millimolar of the persulfate. So here we wanted to try the caffeine concentration. So here you can able to see the 200 milligram per liter showed the maximum rate at 9.28. Whereas if we further increase the caffeine concentration, it started to decrease. So with respect to reaction rate, the 200 takes the much higher edge. But if you think about the caffeine concentration, the 400 ppm, still it can take a much higher rate. So this advantage of utilizing this sulfate radical, which is not experienced by a in the hydroxy radical. The reaction rate was not that much intense with respect to hydroxy radicals. So then the conventional optimization tools we wanted to see, because to make it in an industrial application, we wanted to check with the process control. So the first preliminary advantage for getting into the process control is the model optimization. We wanted to try with the different seasons also. So we tried in response surface methodology as well as the ANN. So here one of the important tool by which you can able to compute what is the maximum I can able to attain the degradation of this caffeine in terms of concentration, not in terms of percentage. And here you can able, and we have incorporated some mathematical calculations also, which is called canonical and ridge modifications. Through this canonical and ridge, we try to understand, SAS programming also we have done, to understand how these models are improvising the data. So which is given in this particular slide. The sensitivity analysis we wanted to establish for the reacting variables with the output variables. So here you can able to see the initial untransformed models gave around 221 ppm, And the canonical ridge also didn't give much revised area. It is almost 221 only. But transformation, when we did, we could able to understand the system much better way up to 244, and optimized condition from CCF is around 290. So actual system works very well for 290, close to 300 ppm of a die can be degraded if I am supplying 500 ppm of a die, and this takes place within 15 minutes of a time. Right, so out of 500, 300 out of 500 in 15 minutes of a time I can able to achieve. So this is what the output. Further, we wanted to try the ANN because we wanted to make it into an industrial application. So they have expressed one important problem. Their effluent treatment plant is like an open system. So northeastern region, as you know that it is having a very good considerable amount of a rainfall, and the humidity is also very intense. So the seasonal variation, how far it is affecting the industrial effluent, we took care of that. So we wanted to try the artificial network model. So by executing almost close to 3000 experiments we have done. So all the 3000 experiments we have executed with the real-time samples, we have incorporated into a model, and we have trained the system for the real-time industrial application. So this is what the data sets. So whatever the data, whatever the samples we have collected from the industry, and we could able to achieve the degradation through sulfate radical systems, and the model what we developed explains it very well at a rate of regression of at 0.97, and you can able to see the differences with the training, validation and test, and the overall R square is also very close to 98. So this is what we have developed for that particular pharmaceutical industry, where they are now we are in the discussion to make it into a pilot scale. Yeah. So, and after getting into a discussion with their MD, we got an interesting factor. We wanted to try because they have used their water content is highly concentrated with the carbonate ions. So as we know that this caffeine degradation works very well in the alkaline pH, right? So we wanted to try how far this carbonate, bicarbonate and chloride ions, which is considered to be natively present at a higher quantity in this northeastern region. So we wanted to try that. So that interestingly gave an results like this is the control where the rate of the reaction is almost to 4.5 ppm per minute. But when I increased the carbonate, it started to increase, the degradation rate is also increasing. So still when the concentration of carbonate is very high in the water sample, native water sample, and the caffeine is present in that, this enhances the efficiency almost 2 ppm per minute as it is increasing. And that is a very interesting factor we got. And then we started to look into the other carbonate, the other form of a carbonate, that is a bicarbonate, so which is considered to be less alkaline than the carbonate. Here you can able to see the difference. It started with 4.6, but it could able to reach only 5.6. But in the stronger one, it could reach up to 6.5, almost 7 ppm. We can able to tell. Here it is almost 6 ppm. 1 ppm was the difference, which is due to the nature of the alkalinity offered by the bicarbonate and carbonate ions. But and very important results we observed from the chloride ion is till 4 millimolar, the chloride ion, if the concentration is there, it keeps on increasing. But beyond 4 millimolar of the concentration of the chloride, the degradation started to decrease. But here the concentration in our water sample is almost close to 6 millimolar. So we are stuck with this particular point, how to reduce this effect of the chlorination. So we are suggesting to dilute the sample so that the process can be much efficient, but still we are working on how to utilize this chlorine to degrade these caffeine in a much better way. So this is what a real-time implementation kind of a work where the real-time samples was taken into study, and the different kinds of reaction kinetics and the conventional route of scientific approach as well as the real-time approach was targeted throughout the study. And here you can able to see the reduction in the carbon, hydrogen and nitrogen. At 0 and 60 minutes, it has reduced all the three, and the higher concentration, you can able to see a drastic decrease in the system, whereas in the higher concentration, nitrogen alone is decreasing. So it is not removing the nitrogen. That is what I have said in the first 3 to 4 slides. I have said beware, if most of the process are targeting on the carbon, we need to target on the nitrogen also. That is a very important one. We have to work on it. And regarding the total organic carbon, you can able to see the reduction in the total organic carbon as well as the caffeine concentration, because we need to map these two. It cannot be like only caffeine concentration. So TOC, which corresponds to 200 ppm, is 82.83 ppm, and it has reduced to 14.4 when the caffeine is reducing to 40.4. So this is how the comparison between the TOC as well as the caffeine was done. So now we got another dilemma, where how to analyze whether the process which we have developed is safe for the environment. So we wanted to study the LCHRMS. So through these data, we have identified what are all the degraded end products, and we have proposed the degradation pathway where the lower molecular weight compounds are coming into the system. So here you can able to see based on the MBZ values and the which is highlighted in the orange and the green one, and the lower molecular weight products till this particular stage we could able to achieve. So the process what we have developed to degrade the caffeine is not yielding any kind of a toxic intermediates, and the proposed pathway for degradation this caffeine towards the lower molecular weights is depicted over here. So this confirms the efficiency of the degradation process to control the caffeine in a much efficient way. And yeah, so this is what I want to, from this point, I would like to conclude the session. And throughout the entire lecture, I wanted to specify one important thing. Whatever it is, whether it is concerned with the pharmaceutical compounds or personal care products, whatever it is, whether it is very specifically to antibiotics or paraben compounds or caffeine compounds, whatever it is, you have to be very careful whenever you need to know how we have to use it. Okay, there will be a prescribed procedure for utilizing it or using it or for consuming it. So please try to follow that particular rules which is given by your doctors or the medical practitioners or medical professionals. Please try to follow that to the point, to the core, so that we can reduce the resistance developed by the microbes against these antibiotics and the pharmaceutical pollutants, so which will delay the process of resistance. Within that time, the other side of the research, that is the antibiotic development or antibacterial agent development or antifungal agent development, researchers will take care of the genesis of new novel drugs. Okay, so the balance has to be there in between these two. If you are not properly handling the antibiotics, definitely it will create a damage with respect to antibiotic resistance. At the same time, you need to be very strong with respect to the systems. Okay, so this is what the major area which we wanted to, and I would like to acknowledge all the funding agencies and our research collaborators for funding this particular, this entire plot of a research with respect to pharmaceutical compounds or textile dyes or pesticides and other related work. And a special special mention about the ministries also, Ministry of Earth Science and DST and DBT for funding these research works to make it into a real-time application, and all other related funding agencies too. And I would like to thank all my research scholars who are working with me and the entire team for which are focused on these particular pollutants in the system. And our major area of research is around the wastewater surveillance through antimicrobial resistance, bioprocessing of the industrial waste, microplastic effect on invertebrates and the symbionts. So these are all the major areas right now we are focusing on. And yeah, thank you. If you need any kind of collaborations, we are always open for the collaborative works. Yes, thank you. If you have any doubts or clarifications, you can. Session is open for that.
Yes, sir. Thank you. Thank you, Professor Padmanabhan.
Yeah.
We have questions in the chat.
Yeah. Okay. Just a minute. I will stop sharing.
Yeah.
Yeah.
Dr. Bhuvaneshwari ma'am has asked one question. If you could see the chat, please.
Yeah, what pharmaceuticals or caffeine is having? The caffeine is added in a very minor quantity in most of the tonics what we are consuming, and as you know that it is a kind of a drug which is widely used to give a neural lift. Okay, so these pharmaceutical systems are adding the caffeine into the toxin. So most of the tonics what we are taking, which gives a natural flavor to the system, so that is having that is added in the system. But the problem with the caffeine is most of the pharmaceutical compounds have a very lesser half-life, but caffeine have a very strong half-life of 10 years. So that is why it is though it is in the PPCPs, it is also classified under the persistent pollutants also. So which enters into the living system, which magnifies, and it enters and creates a lot of toxicity to the living point. Yeah. What are all the antibiotics needed to be treated which are alarming? No, ma'am. This is related to the region wise we can able to put it. Okay, like because if you take any kind of, if you take let us put it in within our India. So here in certain part of a country where the medications are not that much effective, the natural defense mechanism is there. They are dependent on the natural defense mechanism. Wherever in certain part of the areas where we are used to consume the antibiotics for the smaller issues. So the naturally our system gets accommodated into it. So it is a region specific. If you take the south part of an India where the antibiotic usage is very, very high. So which is a very important thing we have to think about it. But if you think about in the northeastern areas or in the Western Ghat region, where naturally the availability of these pharmaceutical compounds are very less. So the scope for the resistance will also be very less there. So it is like a region specific only. Yes, to put it in a different, very precise way, the beta-lactam antibiotics is widely prescribed. Tetracycline, fluoroquinolone antibiotics are highly prescribed. The one important thing is if doctor or physician is prescribing the antibiotics, please try to complete the dose so that it will be completely cleaned, wiped off. Don't stop at the intermittent level. That is a one important suggestion we can be able to give. Yeah, and sir, we want to create a chemical degradation system in small pharma colleges. What we should do? If it is a small, if it is for a research purpose, that is a different thing because that takes a different arena of research. If you want to make it into a real-time application system, you can make it in a complete degradation profiling. So the sulfate radical system is one important area. Otherwise, hydroxy radical is another important one. Fenton reaction is a very important one. These three can be a promising area where if you want to put it in a real-time implication system in a small scale, decentralized way. If you want to take it in research work, then it is a different picture. Now many work are coming. Photocatalytic reactions, or you will be developing the nanoparticle, then you will be putting into the pharmaceutical effluents, then again you will be degrading that. Then again the component of a nanotoxicity comes into the play. Who will take care of the nanotoxicity? So those many different things are there. But so the research perspective is different. We wanted to, it takes more time. How to tackle the nanotoxicity, it is a separate topic of a discussion where the researchers need to emphasize more. But if you want to make it in a smaller level, if you have a huge volume of pharmaceutical waste in your pharma colleges, please try to absorb it and make it into a smaller volume. Once you take it into a smaller volume, use a very strong oxidizing agents which can take care of the degradation. One such oxidizing process is a Fenton's process, sulfate radical process, hydroxy radical process. So these things we can able to experience. What about the role of nanoparticles? Yes, as I said, nanoparticles can be used, but the problem there is, if you see the other side of the nanoparticles, the nanotoxicity is also much important one we have to target. Without understanding the nanotoxicity, if you are putting into the degradation of antibiotics, it will create a much higher impact than the antibiotics as well, because antibiotics can degrade on its own within a period of time, but nanoparticles will be there, will be sustained in the environment for a longer period. And it is having a very important property of photocatalytic activity also. So natural light or UV rays will also elicit the electrons, which also affects the natural living system. So it is not, that is why now the research, if you see the research on nanoparticles is getting decreased with respect to water treatment. The other side of the nanoparticles is there, but towards the water treatment, now the researchers have started to decrease. So that is one of the disadvantages. But what I would say is we need to still focus on the drawbacks of the nanotechnology, like nanotoxicity profiling. We have to target. We have to think about the how these nanoparticles are affecting the toxic organs or tissues of different class of organisms. That has to be extended. If that is extended, then it can be applied in the any kind of a treatment. Yes. Yeah, I hope I have addressed all the questions. Any more queries? If you have, you can ask.
Participants, if you have any questions, you may ask now. So we'll have another 2 to 3 minutes for question session.
So, very warm good evening, sir.
Yes, ma'am. Good evening, ma'am. Good evening.
So, your session was really very nice regarding the antimicrobial resistance. It was a really mind-blowing one. Thank you for the session, sir, for very informative session. Sir, I have a very basic doubt. It is not a very big thing, just a basic question. Many of the doctors are suggesting the antibiotics for 3 to 7 days. If we are having any of the issues, even for the kids or juveniles or for the different age groups, they are suggesting the antibiotics for 3 to 7 days. Minimum duration is for 3 to 7 days. And the highest dose is still about 650 mg they are suggesting.
Yeah, I can understand. I got the crux of your question. The thing is, like, I am not a physician to comment about how much dose you have to take. I am purely an environmental engineer. We will take care of the pollutant which is present in the water. What I would suggest is, whatever the physician says, that is not decided only based on the dealers who are supplying the medicines. It is also depends on the region level resistance which has been experienced by the doctors. So we cannot able to compound, because I personally felt the problem in our, when I came from south to here northeastern region. Here the doctors are prescribing very basic level of an antibiotic for a fever. So what I felt is how this will take care of my illness. Okay, that depends on the region wise also. Here they are, because not much resistant bacteria has been developed, but in the other side of the system it is not like that. So whatever, so the region wise physicians always see to these things and they will prescribe it. Whatever the physician says, you please blindly understand it, get the information from them and please follow it. That is what one suggestion I can able to give.
Okay, sir. So blindly we can believe the physicians, right?
Yes. You can. That is, you can, because whenever you have a doubt, they are bound to explain that also. It is like, it is not like if you are asking like after googling it and you are asking, they will get annoyed. But if you are asking a scientific one, definitely they will be ready to explain the phenomena.
Sure,
So
I can
that
understand.
you can able to use it. Apart from that, I can able to give suggestions on the treatment side. So that if you ask, in most of the rivers of south, we have identified the concentration of the antibiotics in the river streams.
No, what I was trying to ask is whether the gut microbe or the gut microbiome will be getting affected or not. That is the thing I want to know.
Yes, ma'am. Yes, definitely. That is why most of the doctors will also give the one particular capsule which enhances your gut microbes also, if they are giving a heavy dose of a tablets. Heavy dose of an antibiotic, because it is like a we need to control the disease at a shorter time period, otherwise it keeps on spreading. So that is also a possibility for resistant developments. So for considering those important points, they will give a different doses of antibiotics. So that is why,
Probiotics also will be suggested.
yes.
Okay, sir, fine. So thank you so much, sir.
Thank
Might
you,
be
thank you.
it might be a very silly question, and you have
No.
answered that. So thank you so
It's
much.
not like that. All the question, but you have asked me a wrong person. That is why I couldn't able to properly answer. The proper answer means, because since I am not experienced as a physician, I cannot able to comment on that. So that is one thing. Yeah.
Yeah, I can understand. Thank you so much, sir.
Yeah, ma'am. Sir, comparatively, physical or chemical or microbial degradation will be effective. Ma'am, physical process, I will not take it into the component of a microbial degradation, because if you keep on using the microbial degradation, you will be generating the antimicrobial resistant bacteria, which is not good for the environment. So please, one humble request to all the doctorates and all the students, all the scholars over participating over here is, even if you are started your research work on microbial degradation of antibiotics, please try to avoid that. Try to switch over to physical or chemical based treatment. Okay. And when you compare physical or chemical, physical process is energy intensive process and it is effective. Chemical is non-energy intensive, but it is also effective. So by thinking about the energy point of view, chemical treatment or the augmented process will take the upper edge. My vote will be for chemical based treatments and advanced oxidation process.
Good evening, sir.
Good evening.
Yeah, sir, your presentation was good and informative. I have two questions, sir. I'm working on dye degradation. In dye degradation, I have two queries, sir. Number one is how to go with the absorption kinetics. And second is how to do with the reusability of nanoparticles, sir.
First one, adsorption kinetics, you can able to take it in two different aspects. One is to find the order of a reaction, how these adsorption is taking place, at what rate it is taking place. So though that will be like a first order kinetics or pseudo second order, pseudo first order. So that order kinetics will be there. Second one is about the isotherms at a fixed temperature, how the adsorbent and adsorbate is going to interact. Based on that adsorption isotherm, there are many adsorption isotherms are there. Langmuir, Friendlich, Temkin, Voxkener, so many different adsorption isotherms are there. One important thing is it will give you an idea whether it is a physisorption or chemisorption, whether a mere force of attraction is there or any bond is formed. So this is the two different approaches you can able to understand with respect to adsorption. Okay. And what about the second question you have asked? Reusability of the
Reusability of nanoparticles.
If it is only based on the reusability, what I can able to tell is recovery is the major point to reuse the nanoparticles. If you are using the ferrous based nanoparticles, you can able to recover the nanoparticles using magnetic principles. So that is why everywhere the ferrous-based nanocomposites or nanoparticles are MOF or getting developed. The ferrous will be a core component because this ferrous will help you to recover the things will be very easy when you put it into the magnetic system. And another one is about the covalent organic frameworks, where the frameworks will be generated through a metal or through a covalent compound. So these things will be much effective to trap the pollutant in a very confined space. And if you put the light, the excitation of electrons will take care of the degradation. So this is how the recovery is focused as of now. And another approach is through adsorption. You can adsorb through any biomass, you can recover it, but that will be a very cost intensive process. I think nobody will prefer that. Better you can go with the ferrous based nanoparticle, that will be easy for you to recover.
Thank you, sir. Thank you for the information.
Thank you, sir. Thank you, Dr. Ravi Shankar, sir. Thank you. And Ananthi Jayaraman, is there any policy or rules and regulation regarding the? Yes, earlier it was not there, but now the government of India has instructed all the state governments to emphasize the policies and rules. Earlier, I can tell before 10 years of time, there was only 3 biomedical waste or biomedical treatment plants was there in the entire Tamil Nadu state. But now it is not the case. Now each district is having one regulatory body. So now the policies and rules and regulations are very stringent. And now the researchers, the academic researchers, the scientists and the hospital doctors and directors have started to work together towards the pharmaceutical surveillance. antimicrobial resistance, we three have started to work together. So because earlier it was not the case, because you can able to see many hospitals are the point source for the release of antimicrobial resistant bacteria. Now we have identified that, and now we are working on towards the installing the treatment plants in the hospital, so that the treated effluent and the what we can tell the postmortem samples, the biopsy samples, all these are the good carriers of these antimicrobial resistant bacteria, and also the entities also like DNA and RNA. So that we have started to work together, and that is how the recent project we are working on from the Department of Science and Technology on the river streams based hospitals, where how to control these release of AMR into the systems. That is what we are working on right now. Yeah, I hope I have answered you, ma'am. Ananthi Jayaraman, ma'am.
Yeah. Thank you, Badmanabhan, sir. I hope
I have answered everyone's queries.
Yes.
Yes.
So we have come to the end of the session, and I take this opportunity to thank Professor Badmanabhan for his insights and a valuable sharing of his research findings with us. And this would inspire many of us to focus on the research on AMR, antibiotic degradation policy related works. And participants, we are going to wind up our session of day 1 here. So we will start our day 2 sharply by 6 o'clock tomorrow. So kindly join the session before 10 minutes. By 5:50, please join. Our research persons will also get joined there. So hopefully we will have a fruitful session in upcoming sessions also. And if you have any technical queries, you may message us in the in the given numbers. And thank you everyone for the day. Take care. We are winding the session now.
Thank you, Dr. Amar. Okay, bye.